Today we are discussing about double helical structure of DNA. Double helical structure of DNA. This double helical structure of DNA was given by James Watson and Francis Crick. In 1951. And for this structure of DNA, they got a Nobel Prize in 1962. Now, what are the various salient features of this double helical structure of DNA? We will discuss them in detail and one by one. Say, let's take the first salient feature of the double helical structure of DNA, which was given by James Watson and Francis Crick. That is, the DNA which about they were discussing is a right-handed double helical structure. Is a right-handed double helical structure, and this structure is made up of, or it is composed of, deoxyribonucleotides. As we have discussed in our previous lecture, that in case of DNA, we have 4 types of the nucleotides which join together to form our DNA. Like we have deoxyadenylate, deoxyguanylate, like we have deoxycytidylate, like we have a deoxythymidylate. Now these are the various nucleotides which join together and to form a helical structure. That's why the sugar here is a deoxyribose, and we have a ribonucleotides which join together to form our double helical structure. And one more important thing which you have to keep in mind about the Watson and Crick's model, that is a right-handed DNA structure. Second salient feature which you have to keep in mind, that is the 2 strands which are in a double helical structure, they are anti-parallel to each other. That means one runs from 5' to 3' and another runs from 3' to 5'. So the two strands are anti-parallel to each other. They are anti-parallel to each other. That means if we take one strand runs from 5' to 3' and another runs from 3' to 5'. So this is known as the anti-parallel direction. That means you can compare it with the 2 roads. And on 2 roads there is a movement, or 2 parallel roads on which the movement of the traffic is in the opposite direction. Say if we have 2 roads here, and on one side the traffic moves in this direction, and on another side the traffic moves in this direction. So you can compare it with the 2 parallel roads upon which the traffic moves in opposite direction. Let's take this anti-parallel, what does it mean, like this 5', 3'. Let's have the structural view about it. Say first if I am taking a nucleotide here, say this is one of the nucleotide. I am making here a deoxyribose sugar, and we are here at carbon number 1, 2, 3, 4, and 5. On carbon number 5 we have say adenine attached here, and on carbon number 5 we have a phosphate attached here, and we can call this one as a 5' here. And onto this nucleotide we can join another nucleotide. Say if I am joining a nucleotide to it, say another nucleotide to it, say this will be carbon number 1, 2, 3, 4, and 5. On 5, between this 3 and the 5, we have the formation of a phosphodiester bond. Similarly, if I am joining, say there on this carbon number 1, we have the guanosine attached here as a nitrogen base. Now, if I am joining one more nucleotide to it, say 2 phosphates are released as a pyrophosphate, which we have discussed in our previous lecture. So if I am adding one more here, say this is a cytosine here, and then we have the carbon number 1, 2, 3, 4, and this becomes 5, and we have a phosphodiester bond here. Now this is a strand of the DNA. See, if I am taking this strand, it runs from 5' then and we have a 3'. So you can make a double here a helical structure, and this helical structure is here at 5' end. That is carbon number 5 of a pentose sugar, deoxyribose sugar. Similarly, we have this carbon number 3 of a deoxyribose sugar. This becomes a one strand. So if we are continuously adding the nucleotides to it, and you will see on the top we have a 5 dash and on the bottom we have a 3 dash. That is actually the 3 carbon of the deoxyribose sugar. And if we will make its antipolar strand, similarly, what we have to do, we have to make the deoxyribose sugar in this direction. So this is a deoxyribose sugar, okay, and this is carbon number 1, this is a 2, this is a 3, and this is a 4 here, and this is a 5 here. On 5 we have a 3 phosphate tag, but if their nucleotides are joined to each other, then what happens? 2 phosphates are released as a pyrophosphates, and 1 phosphate remains attached here on carbon number 5. Now, see formation here we have, because here we have a guanosine at carbon number 1, because guanosine makes a 3 hydrogen bonds with the cytosine. Similarly, if I am taking one more nucleotide here in opposite direction, if I am taking one more nucleotide in opposite direction, say this is one more nucleotide I am taking here, this will be 1, 2, 3, 4, and this will be carbon number 5. Between 5 and the carbon number 3, we have a formation of a phosphodiester bond. So here is a guanosine. Now on this position we have a cytosine. It again makes 3 hydrogen bonds with it. Similarly, if we take one more nucleotide here in an opposite direction, say this is the one more nucleotide I am making here. This is carbon number 1, 2, 3, 4, and this will be carbon number 5. And between them we have a phosphodiester bond. Now on it we have the presence of thymine at carbon number 1, and it makes 2 hydrogen bonds with the adenine. So if we will see here, what will be the topmost carbon here, that is the carbon number 3, and what will be here, that will be carbon number 5. If I am making a the helical structure of the DNA, now this becomes 3 dash and this becomes 5 dash. So what actually is this 3 dash and 5 dash? These are the actually carbon atoms of the deoxyribose sugar. Say if we will take on this strand, on the top of the strand we have a 5 dash, and that is a 5 carbon of the deoxyribose sugar, 5th carbon, and on the bottom we have a 3 carbon, that's actually the 3rd carbon, 3 dash carbon, that's actually the 3rd carbon of deoxyribose sugar. So that's why we call this one, the 2 strands are antipolar to each other, and these 2 strands, they move in an opposite direction. Now we have one more thing which we have to keep in mind. If we will take these 2 strands, third cell in the future, these 2 strands, they have a diameter or the width of. If we take width or diameter of these 2, the diameter is about 20 angstrom, or we can also call it as a 2.0 nanometer is its diameter. See, if I am making here a double-stranded DNA, see if I am making again a double-stranded DNA here, so I will show you again the structure, what is the width actually here. See, for instance, if I am taking a double-stranded DNA here, say this is a DNA, a double-stranded DNA if I am making roughly. See, this is double-stranded DNA here. Now here we have a diameter between the 2 strands. It is about, say if I am making here, there is also a diameter of between the 2 strands, is about 20 angstrom, okay, or we can also write it as 2.0 nanometer. This is diameter. And one more thing which you have to keep in mind, there is a 4 cell in the future which we have to write about the DNA. That is one turn of DNA, which is also known as a pitch. We can also call that as a pitch. Okay, it is about 34 angstrom. Okay, which is equal to 3.4 nanometer. Okay, or we can also call it as having in this one pitch we have about 10 base pairs. 10 base pairs. So the one pitch, if I make one pitch from this area, if I take from this area up to this area, it's a one pitch. And this pitch is about 34 angstrom, which is equal to 3.4 nanometer. And how many base pairs are here up to this area? From if I am highlighting the red color from this point up to this point, this is one term. In a one term, we have 24 angstrom, its lifetime, it is equal to 3.4 nanometer. So it's about 10 base pairs. 10 base pairs its length. So if you take a double helical structure of the Watson and Francis group, it is known as a BDNA. It is known as a BDNA, and this BDNA is having the 10 base pairs per term. This is the most important point which you have to keep in mind. Now, the more important thing which we are discussing about this one, if we have two nucleotides, say for instance, it is one nucleotide here and this is another nucleotide here, and we have a phosphodiester bond here. The distance between two nucleotides is about is equal to 3.4 angstrom, which is equal to 0.34 nanometer. Is what we call the length or the distance between two nucleotides in a DNA. So if we take two nucleotides who are joined to each other, the distance between two nucleotides will be equal to 3.4 angstrom, which is equal to 0.34 nanometer. Now, one more important thing which we have to keep in mind about the DNA is if we will take a DNA, it is having the hydrophilic backbone, and central, which we have the nitrogen bases which are hydrophobic in nature. Let's see this detailed structure about it. Say for instance, if I am making here again a DNA, a double-stranded DNA, say for instance, if I am making here a larger one, a double-stranded DNA, I show you this backbone. Say for instance, a backbone here. If I am taking here a backbone, it is made up of sugar, then we have a phosphate, then again sugar, then we have a phosphate, sugar, then phosphate, then sugar, then phosphate. On this side we have again a sugar, then we have a phosphate, then we have a sugar, then we have a phosphate, then we have a sugar, then we have a phosphate, then we have a sugar, then we have a phosphate. Okay, now if you take this sugar, say if here we have adenine and here we have a thymine, they make it two hydrogen bonds. If here we have a guanosine and here we have a cytosine, now it makes three hydrogen bonds. Similarly, if on this sugar we have adenine and on this sugar we have thymine, now it makes two hydrogen bonds. Similarly, if on this side we have a cytosine and on this side we have a guanosine, now it makes two hydrogen bonds. That means this backbone which we have, this outer layer, this backbone which we have, it is hydrophilic. Hydrophilic. If it is hydrophilic, it is made up of sugar and phosphate. Sugar and phosphate. The sugar which we have is a deoxyribose sugar. So this portion, this backbone of the DNA, it is hydrophilic because it is made up of sugar and phosphates. If you take this central core, this portion, this central core, it's having the nitrogenous bases, and this central core, it is hydrophilic. It is hydrophobic, so it is water hating. So we can say this hydrophobic portion is consisting of nitrogenous bases. It is consisting of nitrogenous bases. This is the most important thing which you have to keep in mind. That means the backbone of the DNA, which is consisting of sugar, that is a deoxyribose sugar and a phosphate. So it is hydrophilic in nature, that means it is water loving. Because if we will take a cell, say for instance, I am making here a cell. Now within the cell, we have a presence of a nucleus. It is a nucleus in case of eukaryotic cell, and we have a presence of a DNA. Now if it is a double helical DNA here, and it is present inside the nucleoplasm of the cell. Now this nucleoplasm is a liquid matrix. It's having the high concentration of water, about 70%, like we have in the case of the cytoplasm. Now this backbone, it loves to interact with the water. So that is why the backbone is hydrophilic in nature. But this central portion, which is having the nitrogenous bases, it is a core of the DNA. Now this core of DNA is hydrophobic. Core of DNA is hydrophobic, that it heads the water. So that's why it remains within the two strands of this portion. Now we have one more, what we call this challenge feature of the double-stranded DNA. That is, if we will take a Chargaff smooth, if we take a double-stranded DNA, it obeys Chargaff's rule. It obeys Chargaff's rule. What that rule states, that is, the purines are equal to pyrimidines. That means the purines are equal to pyrimidines. That means in case of DNA, we have equal number of purines and pyrimidines, which means we have equal number of adenine on one strand is equal to the thymine of another strand. Similarly, guanine is equal to the cytosine of another strand. So they are equal in number. That means purines on one side and pyrimidines on another strand, they are equal in number. The reason is adenine always makes hydrogen bonds with thymine and guanosine always makes hydrogen bonds with the cytosine. Now the question arises, that is, why this adenine do not bind with the guanosine? Why purine do not bind with the purine? Why it binds with thymine? The reason is, if you take these purines have dicyclic structure. These purines have dicyclic structure. That means there are two aromatic rings. That means the two aromatic rings do not get accommodated into a double-stranded DNA. So if you take this space is not enough for these two structures to get accommodated here. If these two structures are not able to accommodate here, that means the two purines do not bind to each other. Now with one more thing, why not two pyrimidines bind to each other? Like, why not thymine binds to the cytosine? The reason is they are too smaller in structure. The reason is these two are monocyclic. They are monocyclic. That means they are single aromatic ring. That means if we fit them here, the space becomes, these two structures remain far away from each other and they are not able to form a hydrogen bond between each other. That means these two have a monocyclic structure and they remain far away in a double-stranded structure and do not form the hydrogen bonds. And these two structures, these purines, they have dicyclic structure, two aromatic rings. That means there is no enough space so that they can be accommodated here. So the best combination is purines should combine with the pyrimidine because purines have a double aromatic dicyclic ring, whereas pyrimidines have a monocyclic ring. It is there enough. This space is enough for these two structures to get accommodated here as to form the hydrogen bond. So that's why we say the Chargaff's rule states that there is an equal number of purines and pyrimidines in case of a double-stranded DNA. That means on one strand, if we have adenine and thymine equal to that number, equal to 100 units. Similarly, on another strand, the thymine and cytosine will be also 100 in number. So this is most important thing which you have to keep in mind, which was given by the Chargaff. So this B type of DNA, which was given by the Francis and Crick, also obeys Chargaff's rule. Now tomorrow we'll discuss some more points or some more postulates of the double helical structure.